We are preparing for the SeekSpace single-cell spatial transcriptome seq experiment. Now, let's get started. The first step is to prepare our nuclei suspension from the tissue slice and get them labeled with spatial barcodes. To begin tissue labeling, place the spatial chip into the SeekSpace chip holder and secure it with the safety latch. Incubate the assembly in a thermal cycler or a slide dryer according to the user manual to bring the chip to the required temperature before chamber installation. Keep lid open. While the chip is warming, prepare the space chamber by removing the protective films. Once the incubation is complete, align the chamber over one spatial barcode area and gently press around the edges to create a secure seal. Repeat the same procedure for the second spatial barcode area, ensuring both chambers are fully sealed before proceeding. Add the N1 solution to each reaction chamber and gently pipette to ensure the tissue section is fully covered. The labeling reaction transfers spatial barcodes from the chip surface into the nuclei within the tissue section. Place the chip assembly into the thermal cycler and start the labeling program. After labeling, remove the reaction solution and wash both chambers twice with 1 times PBS-RI to remove residual labeling reagents. Finally, add fixation solution to both chambers and incubate at room temperature for 12 minutes. Start the timer immediately after addition to ensure accurate incubation timing for fixation. During fixation, proceed with fluorescence imaging of the tissue. Release the spatial chip from the SeekSpace chip holder by pressing the latch and place it upright on the microscope stage for imaging. Perform fluorescence imaging using the recommended microscope settings according to the user manual. Minimize fluorescence exposure by turning off the laser when imaging is not in progress. Fluorescence imaging should be completed within 12 minutes while the sample remains in fixation solution. If imaging cannot be completed within the time window, replace the fixation solution with nuclei extraction buffer and complete imaging within an additional 10 minutes. This step is time sensitive and must be completed under controlled exposure conditions. Hematoxylin and eosin staining is performed for the adjacent tissue slice. Spatial labeling and imaging are complete. Next, extract the spatially labeled nuclei and assess nuclei quality.